welcome to the deep dive.
Yeah.
Today, we're grabbing some of those foundational high-yield sources, your core textbooks for A&P, micro, histology, and really distilling them down. We want to get to the essential knowledge you absolutely need to master in the health sciences.
And our goal here is all about scientific clarity. We're trying to synthesize these really complex structures, these mechanisms, and lab protocols.
Right.
The point is to move beyond just, memorizing facts. We need to understand the application, the context of this knowledge.
Because in practice, precision is everything.
It is, whether you're looking at a gas gradient or you're prepping a slide.
Okay, so let's unpack this. We've basically broken this down into three big domains. They seem separate, but they're all connected by this idea of precision.
Yeah.
So first, the physics of life respiration.
Yeah.
Then the body's architecture, histology, and finally, the rigor of lab science.
Let's jump right in with section 1 then. The physics of life, specifically gas exchange, the movement of oxygen and CO2, it's not magic. It's driven entirely by these fundamental gas laws. We have to remember Dalton's law.
Which says the total pressure is just the sum of the individual parts, right?
Exactly. The sum of the partial pressures. And that partial pressure is what physically pushes the gases to move. Okay,
and this is where it gets really interesting for me, the gradients. The partial pressure gradients that actually drive the diffusion.
Yes.
So let's look at oxygen, O2. It uses a really large, a very steep gradient.
It does. In the alveoli, the PO2 is, around 105 millimeters of mercury? But the venous blood arriving at the lungs is much lower.
How much lower are we talking?
Often just 40 millimeter Hg.
Wow. So that's a huge difference.
A massive 65 millimeter Hg difference. And that means O2 molecules just flood into the blood, raising its PO2 up to about 100 millimeter Hg by the time it leaves the lungs.
And then when that blood gets to the tissues,
the reverse happens. The tissues are using oxygen, so the PO2 there is low, maybe back down to 40 millimeter Hg, and the oxygen just flows out of the blood and into the cells.
So that's why the range for blood PO2 is so wide, it can be anywhere from 40 to 100.
Exactly. It completely depends on where you are in the circulatory system.
Now contrast that with carbon dioxide, CO2. The PO2 gradient is tiny.
It's remarkably small. Venous blood coming to the lungs is about 45 millimeter Hg. The air in the alveoli is about 40.
So only a 5 millimeter of Hg difference. How is that enough to get all that waste gas out?
Well, the key is solubility. CO2 is significantly more soluble in blood than oxygen is.
How much more?
Roughly 20 times more soluble. Because of that, you just don't need a huge pressure difference to get it to move out of the blood and into the lungs.
Okay, so the mechanism works great. But the clinical significance of CO2, despite that small pressure change, is absolutely immense.
It's a physiological tightrope walk.
It really is.
Completely. Unlike oxygen, the blood PO2 level is very, very tightly regulated. We're talking a window of 40 to 45 millimeter Hg.
And that's because every cell is constantly making CO2.
Constantly. It's a byproduct of metabolism. When that CO2 hits the blood, it combines with water and forms carbonic acid, which then becomes hydrogen ions and bicarbonate.
The bicarbonate buffer system.
The single most important buffer system in the body. It's what keeps your blood pH in that incredibly strict range of 7.35 to 7.45.
So if your PO2 creeps up even a little bit, you start getting acidotic.
And if it drops, you become alkalotic. So maintaining that tiny 5 millimeter Hg window is non-negotiable for keeping your whole body's chemistry stable.
Let's shift from the molecular level to the mechanics, the actual movement of breathing. This is all driven by another physical principle, right? Boyle's law.
Yes, that inverse relationship between pressure and volume. It's the engine of ventilation.
So when you inhale, the diaphragm contracts. The intercostals pull the rib cage up and out.
You're massively increasing the volume of your thoracic cavity, and the lungs expand with it.
And because P1V1 equals P2V2, that bigger volume means lower pressure.
Instantly lower intrapulmonary pressure.
So when you say negative pressure, you just mean it's lower than the air pressure outside.
Exactly. It acts like a vacuum, and air just rushes in to equalize it. Expiration, at least at rest, is the opposite and mostly passive. Muscles relax, volume shrinks.
Pressure goes up, and air gets pushed out.
Simple as that. And we can quantify all of this. We measure lung volumes and capacities, which is so crucial for diagnosing lung disease.
Right. So you have your basic tidal volume, your standard breath, around 500 milliliters.
And then the reserves, the inspiratory and expiratory reserve volumes, IRV and ERV.
But the really key concept for assessment is the capacities, which are just combinations of those volumes.
Yes. Like inspiratory capacity is just your tidal volume plus your inspiratory reserve volume.
But the one that's really crucial to remember clinically is the residual volume, the RV.
The air that's left in your lungs, even after you've forced out as much as you possibly can, it's usually around 1,100 to 1,200 milliliters.
And why is that one so important to flag?
Because you cannot measure it with simple spirometry. A patient can't blow it out, so the machine can't see it.
So if you suspect a disease that affects residual volume,
or any capacity that includes it, like total lung capacity, you have to move on to more complex tests, things like helium dilution or body plethysmography. Knowing what your tool can't measure is half the battle in diagnostics.
that structural thinking is a perfect transition to our second domain, the architecture of the body. Let's turn the microscopes inward and look at histology.
Where structure absolutely defines function.
And we have to start with the core protective layer, the epithelial tissue.
Epithelial tissue is all about covering, lining, and forming glands. Its main job is protection. But the key structural point, the thing you have to remember, is its relationship with the tissue underneath it.
You mean the fact that it's vascular?
Exactly. It has no blood vessels of its own at all.
So all the epithelial cells, they have to get their oxygen and nutrients by diffusion from the deeper connective tissue below.
Yes, from the dermis. It's a trade-off. You get a great barrier, but it has massive implications for how that tissue gets repaired.
It really does.
Yeah.
Now, if we look at that underlying connective tissue, which all comes from mesenchyme, the diversity is just incredible.
It really is. I mean, think about cartilage. It's tough. It's flexible. So important for our joints.
And, crucially, like epithelia.
It's also a vascular. The cells, the chondrocytes, are just sitting in these little isolated cavities within the matrix.
So what's the clinical implication of that? Let's say, compared to skin, which is healing all the time.
The implication is repair. Your skin epithelia are highly regenerative because they're always getting damaged. But cartilage, without that direct blood supply, heals incredibly slowly, if it heals at all.
Which is why a serious joint injury can be a lifelong problem.
That one structural detail dictates the entire prognosis.
And we also see things like dense connective tissue, high tensile strength, tons of protein fibers.
Like in tendons. And if those fibers are arranged irregularly, like in dense irregular CT in the dermis, it gives you strength in all directions. It stops your skin from tearing when it's pulled.
And then you have adipose tissue, which kind of breaks the CT rules.
It does. Most connective tissue is mostly matrix, not many cells. Adipose is the opposite. It's highly cellular and highly vascular.
It looks empty on the slides, right?
Because the fat droplets get dissolved during the staining process, but that high vascularity is key. It lets it store and release energy very, very quickly.
For a really complex example of organization, we have to look at the liver. You have to understand the hepatic triad.
You do. The triad is 3 vessels that always travel together at the corners of a classic liver lobule. You have a branch of the hepatic artery,
Which is smaller, with a thicker
a
wall,
branch of the hepatic portal vein,
larger, thinner wall.
and a bile duct, which is lined with that simple cuboidal or columnar epithelium.
But the truly genius part of the structure is how it collects bile. The liver is filtering blood, but it's also making this product, bile, that can't be allowed to mix with the blood.
And it doesn't. It's a completely separate system. You have these tiny channels called bile canaliculi.
And they're not separate tubes.
No, they are literally formed by specialized grooves between 2 adjacent hepatocytes. The cell membranes of 2 liver cells form the wall of the channel.
That's incredible. So bile is collected and segregated from the blood immediately.
An immediate, perfect separation system built right into the cells.
This also gets at why histologists have different ways of looking at the liver lobule, the classic model versus the portal lobule.
Right. The classic lobule puts the central vein in the middle, so it's emphasizing the direction of blood flow.
Okay.
But the portal lobule is a functional concept. It puts the hepatic triad at the center and emphasizes bile drainage. It's a different way of thinking about the same structure, depending on whether you're focused on blood supply or, say, a pathology related to bile production.
This relentless need for precision, whether we're talking physics or anatomy, leads us right to our third section, the rigor you need in the lab.
Absolutely. Our conclusions are only ever as good as our methods.
So controlled experiments,
No.
isolating that one variable.
It's the only way to be sure your experiment actually caused the result you see. And to do that, controls are completely non-negotiable.
We've got 2 main types, the positive control and the negative control. So what happens if my positive control fails?
If your positive control, the one you set up specifically to give you a known positive result, fails, your entire assay is invalid, period.
So you just stop.
You stop and you troubleshoot. It means your reagents are bad, your machine isn't working, something is wrong with your technique. The experiment could not have worked no matter what the other samples look like.
And the negative control, that's what establishes your baseline. It's set up to show no change.
Yes. So if you're testing an antibiotic, your negative control is the bacteria growing happily without any drug. If you see some weird contamination or growth on that negative control plate,
You know you have a false result somewhere else.
it's an essential check against both false positives and false negatives.
In microbiology, this precision also comes down to the media you use. So let's quickly define two key types.
Okay, so a selective medium has an inhibitor in it. It's designed to stop or slow down the growth of stuff you don't want, letting only your target organism grow.
And a differential medium.
A differential medium is different. It's made to detect biochemical differences between organisms. It usually has some kind of pH indicator that causes a color change.
And we use these all the time when we're dealing with enteric organisms.
We do, and the definitions here really matter. An enteric is a broad category. It's basically any gram-negative rod from the gut that ferments glucose.
But a coliform is much more specific.
Much more specific, it's a subset of those that produces both acid and gas from fermenting lactose. And that distinction helps you rapidly identify things in clinical samples or, say, test water quality.
So once we have data from all these rigorous methods, how we visualize it is just as important.
It determines whether your conclusions are even valid. If you're comparing two variables, you have to use an XY scatter plot, and the axes are sacred.
The independent variable, the thing you control, like time or dose, always goes on the x-axis.
Must go on the x-axis. And the dependent variable, the thing you measure in response, must go on the y-axis. If you swap them, you completely reverse the cause and effect relationship you're trying to show. It becomes nonsense.
When you have your data plotted, you don't just connect the dots.
Never. You use a best fit line. It's a line that mathematically shows the overall trend of all the data points together. Connecting the dots suggests a perfect relationship that almost never exists in biology.
The most practical use of this has to be the standard curve.
Or a calibration curve, yeah.
Yeah.
It's the absolute backbone of quantitative lab work.
So you plot known amounts of something on the x-axis against the results you measure on the axis.
Right. You create a known relationship. Then you can take your unknown sample, measure its Y value like its color intensity or absorbance, and use your best fit line to trace back and find its true X value, its concentration. It's pure analytical precision.
Okay, let's wrap up with one last truly fundamental skill where precision is everything. Pipetting.
Oh, yeah. Tiny errors here become huge errors later on.
So rule 1, always read the bottom of the meniscus.
Always. And you have to know your equipment. A serological pipette is calibrated to deliver, or TD. That means you have to blow out that last drop to get the full volume.
Whereas a Mora pipette is different.
Totally different. It's not graduated all the way to the tip. You have to stop the flow right at a specific calibration line. No blowout. If you mix them up, your measurements are instantly wrong.
Okay, so to pull some of these ideas together, especially the physiology, here's a question for you to think about.
Let's hear it.
We said the blood PA2 level varies really widely, between 40 and 100 millimeter Hg, reflecting oxygen delivery and use. But the PiCO2 level is held in that super tight 40-45 millimeter Hg range. So the question is, why is that PiCO2 range so physiologically narrow? And what key chemical buffer system depends entirely on that precise regulation?
That's a great one to chew on. That was a really rapid tour, but it hits on the core ideas that ground A&P, micro, and lab work.
The big takeaway really is that precision in observation is the ultimate skill you're building. Whether you're analyzing a tiny gas gradient, or spotting in a vascular tissue on a slide, or just making sure your positive control actually worked.
And to leave you with one final thought, let's go back to that structural precision. We talked about cartilage being a vascular. It's essential for our joints, but it has no blood supply.
Yeah.
All its nutrients have to diffuse slowly through the matrix.
Right.
So think about this. What might that chronic lack of a robust blood supply imply about our ability to use drugs or even genetic therapies to speed up the repair of damaged cartilage, especially compared to a highly vascular tissue like bone or muscle? That's a huge area of ongoing research for you to explore.